Welcome back to another in-depth training video. Today we will be examining both neuron and cardiac action potential and looking for the differences between them. Both neuron action potentials and cardiac action potentials involve electrical signals. They have distinct characteristics due to their different functions and the types of cells involved. First, we will dig into the cell types. A nerve cell, also known as a neuron, has a specialized structure that enables it to transmit electrical and chemical signals throughout the nervous system. Here are the key components of a typical neuron. Cell body, soma. The cell body contains the nucleus and most of the neuron's organelles. It's the metabolic center of the neuron and integrates incoming signals from dendrites before transmitting the signal through the axon. Dendrites. These are branched extensions protruding from the cell body. Dendrites receive signals from other neurons or sensory receptors and transmit these signals toward the cell body. Axon. An axon is a long, slender projection extending from the cell body. Its main function is to conduct electrical impulses, known as action potentials, away from the cell body toward other neurons, muscles, or glands. Axons can be covered by a myelin sheath, which insulates and speeds up the transmission of the action potential. Myelin sheath. Some neurons are enveloped by a myelin sheath, which is made of specialized glial cells, oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system. This fatty substance provides insulation and helps in the rapid transmission of nerve impulses along the axon. Nodes of Ranvier. These are small gaps in the myelin sheath along the axon. They play a crucial role in allowing the action potential to jump from one node to another, which accelerates the conduction of nerve impulses. Axon terminals, synaptic terminals. At the end of the axon, there are numerous small branches called axon terminals. These terminals form synapses, which are junctions with other neurons, muscle cells, or gland cells. Neurotransmitters stored in synaptic vesicles within the axon terminals are released into the synapse, transmitting signals to the next neuron or effector cell. Synapse. This is the junction between two neurons or between a neuron and an effector cell, such as a muscle or gland cell. The presynaptic neuron releases neurotransmitters into the synaptic cleft, which then bind to receptors on the postsynaptic membrane, transmitting the signal to the next cell. Neurons come in various shapes and sizes depending on their functions within the nervous system. Their structural specialization allows for efficient communication, processing, and transmission of information throughout the body. Next, let's dig into the cardiac cell. Cardiac cells, also known as cardiomyocytes, are the specialized cells that make up the heart muscle. They have unique structures that allow the heart to contract rhythmically and pump blood effectively. Here are the key components of a cardiac cell. Cell membrane, sarcolemma. Similar to other cells, the cell membrane, or sarcolemma in muscle cells, surrounds the cardiomyocyte, separating its interior from the extracellular environment. It regulates the passage of ions and nutrients in and out of the cell. Myofibrils. Cardiac cells contain myofibrils, which are cylindrical structures composed of contractile proteins called actin and myosin. These myofibrils are organized into repeating units called sarcomeres, which are the fundamental units of muscle contraction. Sarcomeres. Sarcomeres are the functional units of muscle contraction and consist of actin and myosin filaments arranged in a specific pattern. The interaction between these filaments during contraction generates the force necessary for muscle contraction. T-tubules, transverse tubules. T-tubules are invaginations of the cell membrane that penetrate into the interior of the cardiac cell. They help transmit action potentials deep into the cell, ensuring synchronous contraction of the entire cell. Sarcoplasmic reticulum, SR. The sarcoplasmic reticulum is a specialized type of endoplasmic reticulum found in muscle cells. It stores and releases calcium ions, Ca2+, which play a crucial role in triggering muscle contractions. Calcium release from the SR initiates the interaction between actin and myosin, leading to muscle contraction. Intercalated discs. These are specialized structures that connect adjacent cardiac cells. Intercalated discs contain gap junctions, which allow for the rapid spread of electrical impulses between cells. They also contain desmosomes, which provide mechanical strength and prevent cells from pulling apart during contraction. Nucleus. The nucleus of the cardiac cell contains genetic material, DNA, and regulates the cell's activities, including protein synthesis and cell function. Cardiac cells are highly organized and interconnected to form the myocardium, the heart's muscular layer. The coordinated contraction of these cells, initiated by electrical signals, allows the heart to pump blood effectively, maintaining circulation throughout the body. But what about their functions? Neuron action potential. Neuron action potentials are involved in transmitting signals over long distances within the nervous system. They facilitate communication between neurons and are crucial for sensory perception, movement, and cognitive processes. Cardiac action potential. Cardiac action potentials coordinate the rhythmic contractions of the heart. They are responsible for ensuring the synchronized contraction and relaxation of the heart chambers. atria, and ventricles to pump blood efficiently. Both action potentials have very different phases. It is important to identify the differences and similarities between these phases. Neuron action potential. Here's a simplified breakdown of the stages of an action potential. Resting potential. At rest, the inside of a neuron is negatively charged compared to the outside. This difference in charge is maintained by the sodium-potassium pump and ion channels in the neuron's membrane. Depolarization. When a stimulus reaches the neuron, it can cause the membrane's voltage to change. If the stimulus is strong enough, it triggers the opening of voltage-gated sodium channels. This allows sodium ions to rush into the cell, reversing the membrane potential from negative to positive. This phase is known as depolarization. Peak potential. As the membrane potential becomes more positive, it reaches a peak value called the action potential. This peak is reached due to the influx of sodium ions. Repolarization. Following depolarization, potassium channels open, allowing potassium ions to move out of the cell. This outward flow of positively charged potassium ions helps restore the cell's negative charge, causing repolarization. Hyperpolarization. Sometimes the membrane potential briefly becomes more negative than the resting potential. This phase is known as hyperpolarization and occurs because the potassium channels remain open for a short period after repolarization. Refractory period. After an action potential, there's a brief period called the refractory period when the neuron is less responsive to additional stimuli. This helps ensure that the action potential moves in one direction along the neuron and allows the neuron to recover before firing again. You can pause here to take notes. Next, we will dig into cardiac action potential. The cardiac action potential differs from the action potential in neurons. Cardiac cells have a specific pattern of depolarization and repolarization that is essential for the heart's function. There are several phases in the cardiac action potential. Phase 0, rapid depolarization. This phase involves the rapid influx of sodium ions through voltage-gated sodium channels. This influx of positive charge causes the cell membrane to depolarize rapidly, initiating the contraction of the heart muscle. Phase 1, initial repolarization. Following the rapid depolarization, there's a brief initial repolarization due to the transient opening of potassium channels, allowing some potassium ions to leave the cell. Phase 2, plateau phase. Unlike neurons, cardiac cells have a plateau phase where the membrane potential remains relatively stable. This phase is primarily sustained by the influx of calcium ions balanced by some outward potassium flow. Calcium ions entering the cell help in muscle contraction by triggering the release of more calcium ions from the sarcoplasmic reticulum. Phase 3, rapid repolarization. In this phase, voltage-gated potassium channels open further, allowing a large efflux of potassium ions out of the cell. This causes the membrane potential to rapidly repolarize back to its resting state. Phase 4, resting potential. The cell returns to its resting membrane potential, ready for the next action potential. Pause the video to take notes here. Next, we will look at the sodium-potassium pump for each action potential. Sodium-potassium pump in resting state. The pump utilizes ATP, adenosine triphosphate, the cell's energy currency, to transport ions. It is an active transport mechanism. At rest, the sodium-potassium pump actively transports 3 sodium ions, Na+, out of the cell, and 2 potassium ions, K+, into the cell against their respective concentration gradients. Neuron action potential. Before an action potential, the resting membrane potential is maintained by the sodium-potassium pump, establishing the ion concentration gradient essential for the generation of action potentials. The pump continuously works to restore the ion gradients after an action potential, contributing to the repolarization phase by extruding excess sodium ions and bringing in potassium ions. Cardiac action potential. Similar to neurons, the sodium-potassium pump is essential for maintaining the resting membrane potential of cardiac cells. It helps reset ion concentrations during phases of the action potential, contributing to repolarization and ensuring the proper sequence of ion movements during contraction and relaxation. In summary, the sodium-potassium pump plays a fundamental role in establishing and maintaining the ion concentration gradients necessary for the generation and propagation of action potentials in both neurons and cardiac cells, contributing to normal cellular function and electrical signaling in these cells. Understanding these differences is crucial in grasping how neurons transmit signals throughout the nervous system and how the heart maintains its rhythmic beating through coordinated electrical activity in cardiac muscle cells. Thank you for watching our videos. Please like, share, and subscribe when you get a chance.